can cut it up, but really what we want to talk about. I know. It's also not working with me at all. Oh, it is. Okay. So I really want to talk about something called restriction enzymes, which is the most important thing for DNA technology. Everything else we're really going to kind of walk through. Okay. So in order to manipulate DNA, in order to have bacterial cells make more of a product that we need, whether it's a growth hormone, human growth hormone, or human insulin, whatever it may be, we can use an enzyme called restriction enzyme. The goal of a restriction enzyme is to cut DNA. And restriction enzymes cut DNA at specific sites. So they will read a certain sequence of codes. So let's say maybe it's A, C, T, G, C, or something like that. It will recognize that and it will cut it. When you cut two pieces of DNA with the same restriction enzyme, you can glue them together. Why? Because what it does is it makes a cut with something called a sticky end. So it cuts it so that one side is a single strand and one side is a double strand. So restriction enzymes cut, they're found in bacterial cells. And then because these restriction enzymes were found in bacterial cells, DNA technology advanced all because of this one finding. Okay, so I really want you to know what restriction enzymes are and how important they are in DNA technology.
Wait, so it lets two separate strands get glued to each other, or what gets glued to each other?
Different pieces of DNA. So like a human one with a bacterial DNA, right? So how do I put in a human DNA into a bacterial one? We cut them with the same restriction enzyme. I'll show you what it looks like in a minute. And then we're going to glue them together. Okay. So here's how this goes. There's something called a restriction site. If you are related, your restriction sites are similar. This is how they do paternity testing. This is how they do crime scene analysis. It's by these restriction enzymes. So restriction enzymes have restriction sites. These are sequences on the DNA that the restriction enzyme can cut. Again, there are certain restriction sites that are common just if you're related to each other, and certain ones that are not common, that are common just in the population. Those are not the restriction sites we would look for if we're doing paternity testing. If you're doing paternity testing, you look for restriction sites that are within that family, right? And if you look to see if they're similar, then you know they're related. Yes?
How would you look for that?
It's called DNA gel electrophoresis, which we'll look at in a minute. Yeah.
I understand why we use it to look at things like that, but what's the point of it? Why does it want to?
Well, okay, so we don't have a reason for restriction enzymes, but bacteria, that's their defense mechanism for foreign DNA that comes in. It's a way of chopping it up. That's how it was discovered. Scientists were studying bacteria and realized bacteria have this great enzyme that chop up DNA. And then throughout the years of studying this enzyme, they realized it can be used to clone genes. It can be used to do a whole bunch of different things.
And these aren't the hydrolytic enzymes, right?
No.
Yeah. So these enzymes are bacteria?
No, these enzymes are found in bacteria. Okay?
Wait, so this is bacteria's DNA?
No, any DNA can be cut with the enzyme because all DNA is read the same. A, C, T, G. Doesn't matter who the organism is. Does that make sense? Okay, so some of the restriction enzymes cut where it's just a blunt end, but that's not very useful for us in this DNA technology. Now, this is what I was talking about. It cuts and makes what is called a sticky end. So DNA is double-stranded, right? And then it chops it up so that the ends are single. Okay? So when you have single-stranded ends, it can go and bind to one that's complementary. So what that means is if I have bacterial DNA and I have human DNA and I cut both of them, with the same enzyme, it'll have complementary sticky ends, so I can insert the human DNA into bacterial DNA. Yes?
Would you say that the single string ends like in what? They said
Sticky ends. The single stranded can bond to the complementary bases. This is very important. This is the most important thing in DNA technology that you need to know and remember. Everything else that I talk about, I'm not testing you on. This I want you to know. Okay? I'm not going to test you on cloning and how cloning is done. I'm not going to test you on a bunch of other things. But I will ask you about restriction enzymes and what the purpose is. Yes? Right. So what are restriction enzymes? What is the purpose? What is the restriction site? Yes.
wait, so the sticky ends, what's it bond to different bases?
Not different bases. It will, so when you cut with the same restriction enzyme, right? If I cut human DNA with a restriction enzyme, I cut bacterial DNA with a restriction enzyme, with the same restriction enzyme, they will have complementary sticky ends. So this way I can insert a human DNA into bacterial DNA, so that I can manipulate bacteria to make more of the product I want. So let's say I want human growth hormone. Where am I going to get human growth hormone? I want a lot of it. The only way is to get an organism to make it. How would we make it outside of the human body if we need more of it? We're manipulating bacterial DNA to make human product. That's the whole point, right? You're telling the bacteria, okay, here's a gene. The bacteria is going to read it the same way that we're going to read it, right? The AUG is methionine and so on and so forth. And once it does, then it can make the product that goes with it. This one? Yes. We don't make them. This is all bacterial enzymes. And they do that so that they can chop up foreign DNA. It doesn't necessarily have a use for us, but it happens. Right. So you manipulate bacterial DNA to make human product. Yes.
What's stopping the two sticky ends from just going back together?
Nothing. Good point. This is why when you do what's known as transformation, it fails a lot and works a very small percentage of the time, because it does exactly what you just said. It goes back and puts itself back together the way it was. No, bacteria don't have that. No, restriction enzymes occur naturally in bacteria, but us manipulating it in order to have bacteria do stuff, that's all lab, right? DNA technology is really just all done in the lab. Okay. All right. So this is what it looks like. So if we cut it, and EcoR1, that's just the name of an enzyme. It doesn't matter. You don't need to know it. But there are different enzymes named for different things based on where they're found and from what kind of bacteria. So when we cut it with an enzyme, with a very famous one that's used a lot, EcoR1, it chops it up so that it has these ends, right? And then when you have those ends, now they're sticky ends. So you see how it says gene of interest? That means that's the gene I want to take out and put it somewhere else, wherever that's somewhere else. It can be human. It can be, I don't know, some other organism's gene that I want to amplify or make a product of, whatever the case is. By having restriction enzymes cut it with sticky ends, and having the sticky ends have complementary ends, obviously, right, because they're single ends there, and then you can just glue them on top of each other. Again, it doesn't always work. This isn't a perfect world, okay? So then once we cut it, I'm going to go through this quickly, and I'm just going to show you what it looks like. We do something called gel electrophoresis. What that is charge and it has a size, right? This is a piece of gel that has little pores in there. We run electric current through the gel, and we have the pieces of DNA move through. So this we do in paternity testing, crime scene analysis, and so on. So forensic scientists do this all day long. You take the piece of DNA from, let's say, blood you found on the crime scene, right? You take that DNA, you chop it up with a restriction enzyme, and you run it through. And you get bands, because they'll give you different sizes based on where these restriction sites are. Now, let's say you have a suspect, and you want to know if that blood that you found on the scene matches the suspect. What you do is the same thing you did. You take their DNA, you cut it with the same restriction enzyme, you run it through, and you compare bands. If you have a bunch of bands that match up, what does that tell you? That's the person that the blood came from, right? Now, we're trying to match up the bands that are not very common in the population, right? There'll be certain sizes of DNA that will get cut up just because those sites are very common. Now, if I have a restriction site that's found 1 in 10 billion, and there's only 8 billion people on Earth, you know that if those two match up, there's some kind of relation there, right? But if I'm looking at a restriction site that's found 1 in 1,000, that's not a good comparative tool, right? So we try to find the ones and match the ones that are less common. And then when you get like a anytime crime analysis or paternity testing, they'll tell you with 98% certainty, right? There's no 100% certainty in any of these because you can still be wrong. Yeah. Many. Why? Because of the sequencing. Remember we said junk DNA? That 90% are these extra, well, that's where all these restriction sites are. Yes. Yeah, exactly. Because you take DNA from parents, and so you have similar restriction sites based on relation. Yes. What do you mean, how do you take DNA? Swab from the cheek is how you get your cells, right? Cheek cells. And then what they do is they break, there's a chemical you put, it breaks open the cells. Then you centrifuge, you spin, you do a whole bunch of things, and you get the DNA. And then you chop up the DNA, and then you run the gel. Yeah. Yep, exactly. It's the best match. So at the end, they say with 98% certainty. So, well, there's still that 2%, right? Repeat that. The pieces of DNA, yes, that are cut by the restriction enzyme. Yes, exactly. DNA fragments are pieces that are cut by the restriction enzyme. Know that it's run through a gel. Okay, so once we should know restriction enzyme, we should know what they do. They chop up DNA, and then how do you do a comparison of DNA is based on this very, really cool technology called DNA gel electrophoresis. Let me see if it's here.